Welcome back, guys. We are moving on into complex inheritance patterns, which is concept 2 in our heredity unit. So just reviewing Mendel's laws really quickly from concept 1, you have the law of dominance, which are saying that some alleles are dominant and some alleles are recessive. If you have a dominant allele, it's going to overshadow the recessive allele. Then you also have the law of segregation that said organisms inherit 2 copies of each gene. One comes from each parent, so you only pass 1 copy of each gene on in your gametes. And then the law of independent assortment, which is saying that genes for different traits are going to assort or assemble differently and independently of each other when your gametes are forming. So you don't have to give the same combination of genes or alleles in each one of your gametes, your sperm or your eggs. So what this means, it boils down to the idea that there is also a chromosome theory of inheritance. So we know genes are located on chromosomes, and the behavior of chromosomes during meiosis accounts for a lot of your inheritance patterns. So there are some exceptions to Mendel's laws. So some alleles are neither dominant nor recessive, and there are several traits that are controlled by multiple alleles or multiple genes. So think about your own traits. What do you already know that doesn't fit within Mendelian genetics? So what traits can you think of to where you don't just have this or this, where you have more than 2 options? You could come up with a lot of different things. So eye color, hair color, height, skin color, all of those things is not just either this or this, right? It's not you have blue eyes or green eyes. There's more than 2 options. So we're going to look at a couple of examples of how that comes about. So our first one is incomplete dominance. So this means that when you have a heterozygous, so remember heterozygous means 1 dominant allele and 1 recessive allele. So when you have that option, or just 2 different alleles really here, neither one of them are dominant or recessive. So when you are heterozygous, your phenotype is somewhere in between. So neither allele is completely dominant or recessive to each other. So for example, if I crossed a red flower with a white flower and the offspring had 1 red allele and 1 white allele, neither one of them completely is dominant over the other one. So instead it mixes or it blends. So the blend of white and red would be pink. So let's look at a practice example on this one. So in humans, curly hair is incompletely dominant to straight hair. So here it's giving me how to kind of notate these on my Punnett square. So it's saying that curly hair is just a regular H and straight hair is an H with an apostrophe on it. It doesn't really matter how you notate these as long as you just make them different so you can distinguish them. The reason why you're using capital H's here, or why they're using capital H's in this scenario, is just so that you know neither one of them is recessive to the other one. So the heterozygous individual has wavy hair. When you have a heterozygous individual, it is that combination between the 2. So wavy hair is kind of in the middle between curly hair and straight hair. So cross 2 people with wavy hair. So a wavy haired person is going to have 1 curly haired allele and 1 straight hair allele. So once again, 1 curly hair allele, 1 straight hair allele. So then I fill this out just like I do a normal Punnett square. So I get these options. Okay, so I have 1 out of 4 boxes, or 25% of this, which is curly hair. Then I have, of my heterozygous, 2 out of 4 boxes, which simplifies to 1/2. And remember, the combination is wavy hair. And then finally I have 1 out of 4 boxes, which is straight hair. So on these, just be very careful and intentional about reading your prompt and knowing what notation that you're using. If it's not directly spelled out for you in the prompt, I would make myself a little note as to what I am using for which allele. So another example. Fitz loves growing flowers for his friend Olivia. Her favorite flowers, roses, are found in red, blue, and purple, which is a mix between red and blue. What would happen if Fitz crossed a blue rose, so blue is this, with a purple rose, which is the combination, one of each. Okay, so I put those 2 parental roses on my Punnett square, and then I fill it out. Okay. So I'm looking for boxes that look like this, and none of them do. So I'm not going to have any options to have red flowers. As far as the heterozygous option goes, I have 2 out of 4, which simplifies to 1/2, which is purple. And then 2 out of 4, which simplifies to 1/2 of being the blue flowers. Okay, so on to our second different type of complex inheritance, which is codominance. So remember, co means like together, as equals. So this means that both traits are fully and separately expressed. So instead of red and white flowers make pink flowers, if they are codominant, it's going to say red and white flowers are going to make red and white speckled flowers. So it's going to be both red and white at the same time. They're not going to blend. They're both going to show up. Another example of this is the blood type AB, because it is showing both blood type A and blood type B at the same time. They're both expressed. It's not blended to make a third option. And then we'll come back to blood type later, but blood type is also an example of something called multiple alleles. So let's do some practice with codominance. So in horses, brown horses are codominant to white horses. So I like to use different letters in this situation to help me out here. You can also use different letters in the incomplete dominance problems, but you just got to pay attention to what type of problem that you're doing. So brown horses are codominant to white horses. The heterozygous phenotype is an Appaloosa horse, which is a white horse with brown spots. Cross a white horse with an Appaloosa horse. Okay, so I have my white horse and my Appaloosa horse, and I cross those to get my different options. Okay, so my different phenotype options, I have brown, I have Appaloosa, and I have white. So none of the boxes are going to be brown. Appaloosa have these 2 boxes, so 2 out of 4, which simplifies to 1/2. And then white, these 2 boxes, which simplifies to 1/2. So when we were talking about blood type, we talked about the fact that it is codominant, and that is because blood type AB is both fully type A and type B at the same time. However, blood type is also something called multiple alleles because there's 3 different allele options. I have the allele A, I have the allele B, but I also have a third option, I. So there are 4 different blood types, which are represented by the phenotypes here. So you have blood type AB, blood type A, blood type B, and blood type O. The genotype that codes for those blood types are listed right beside it. So there's only one genotype that can code for AB because I have to have one A allele and one B allele. There's also one genotype that would code for blood type O. O is a recessive blood type, so I have to have 2 recessive alleles, which in this case is represented by that lowercase I. Notice that A and B are both capital, so we know that means that blood type A and blood type B are dominant. So that means that blood type A, I can either have 2 A alleles or one dominant A that's going to overshadow the recessive I allele. And then same thing for blood type B. I can either have 2 dominant B alleles or I can have one dominant B allele that overshadows the recessive I allele. So this is not something that I'm going to test you on, but especially for those of you all who are interested in the medical field or just want to know how your blood works, I'm just going to quickly go through this so you know what your blood type means. So if this is your blood type, this would be, saying, hey, I have blood type AB, I have blood type A, I have blood type B, or blood type O. So what that means is that you have these proteins that are on the outside of your red blood cells. So these are red blood cells, and these little squares are the proteins called agglutinogens that are on your red blood cells. So if you are blood type AB, you have both the A and the B proteins on the outside of your red blood cells. If you are blood type A, you only have the type A. If you're blood type B, you only have the type B proteins. And if you are type O, that means you don't have either. You don't have any proteins on the outside of your red blood cells. So why this is important is because your body makes antibodies. So antibodies are going to be basically attacking things that don't belong. So what that means is if your body has type A and B proteins, then they're not going to attack A or B proteins. You're not going to have any antibodies because your body normally has A and B. If you have A proteins, that means you don't have B proteins, so your body will attack B proteins. If your body has B proteins, that means you don't have any A proteins, so your body will attack A proteins. So remember, anti kind of means like against, so that's where that's coming from. And then with blood type O, you don't have A or B proteins, so your body will attack both A or B proteins. So this means that you can only receive certain types of blood. So if you are blood type AB, you can literally receive whatever because you do not have any antibodies, so your body's not going to attack anything. So you can get blood type A, blood type B, blood type AB, or blood type O. If you're blood type A, you can only receive A and O because your body has anti-Bs. So anything that would have a B on it, you can't get because your body will attack it. And so as we can see, here's a B and here's a B. So you can't receive blood type A, B, or blood type B because both of those would have the B protein on it. So vice versa with blood type B, you can't have anything that would have the A protein on it because you have anti-A antibodies that will attack it. So you can only receive type B or type O. You can't have AB because AB has A, and you can't have blood type A because A has A. And then finally, blood type O, since you have anti-A's and anti-B's, you can't have anything with A's or B's. So unfortunately that means you can't have blood type AB, you can't have blood type A, and you can't have blood type B. So the only thing that you can accept is blood type O. So that's why O is known as the universal donor, because it can give to everyone. And blood type AB is the universal receiver. I guess I spelled that right. Because it can get any of the other blood types. So this is a different gene than being positive or negative. So your blood type, you might be AB positive or O negative. The positive and negative is for a different protein that is a lot easier because it's just a simple Mendelian dominant and recessive type situation, more so than your blood type here being codominant and multiple alleles. Moving on. So let's look at an example of how to do blood types in a Punnett square. So we have some background information. If a mother has type AB blood, so remember there's only one option for that. I have to have one A allele and one B allele to have AB blood. So there's only one genotype option. And a father has type B blood. Remember with type B blood, I can either be this or this. What are the possible blood types of their children? Since dad has two options and I'm not told which one is the correct one, I have to do two different Punnett squares. One as if he is BB and one as if he is BI. Okay? In both cases, mom only has one option, so that stays the same. And I would fill these out like so. Okay, so in this first scenario, there is a 50% or 1/2 chance of the child having blood type AB and a 50% chance or 1/2 chance of the child being type B. Those are my only two options. If dad is heterozygous for his B blood, meaning he has one B allele and one I allele, then I have a lot more options. I have a 1/4 or 25% chance of the child being AB, this option here. I have 2 out of 4 or 1/2 option of the child being blood type B, because remember both BB and BI code for blood type B. And then I have this third option. So I have a 1/4 or 25% chance of the child having blood type A. So here is another example. Once again, I encourage you to pause and work through this on your own, and then come back and see if you are on the right track with this. Alright, the hospital has made a big mistake and lost track of which baby belongs to each set of parents. This used to happen a lot more frequently than what you would think. That's why the babies have to have those little wrist and ankle bracelets now, actually. So, can you help them fix this before they get sued? So I have 3 babies. Baby 1 is blood type O, baby 2 is blood type AB, and baby 3 is blood type A. And then I have 3 sets of parents. I have Mr. and Mrs. Allele, which both are blood type AB. I have Mr. and Mrs. DNA, which one of them is blood type O and the other one is blood type AB. And then I have Mr. and Mrs. Gene, which is blood type B and blood type O. So even though I have only 3 sets of parents, I have 4 Punnett squares that are given to me. And that's because we know that some blood types have more than 2 genotype options. So let's start off with the alleles. So the alleles are blood type AB and AB. AB is one of the ones that only has one genotype option, so I can feel very confident that this is their Punnett square. Okay. Mr. and Mrs. DNA has blood type O and AB. So AB only has one option, and O only has one option. So since both parents only have one genotype option, I only have to do one Punnett square, and it's this one. So then I have Mr. and Mrs. Gene, and once again I have blood type O, which only has one option, but the other parent is blood type B, and if you remember, B has two options. It can either be BB or BI. So I have to do two Punnett squares to come up with all the different options for this one. Alright, great. So now I'm going to look at my babies and see where I can find each one of these. So baby one is type O. The only type O babies that I can see are right here. So because of that, I know baby one has to be Mr. and Mrs. Gene. Okay, so baby two is type AB. And looking at my two Punnett squares here, this is the only time where I have AB. So baby 2 has to be the alleles baby. Then finally baby 3 is blood type A. So I have blood type A here, so I know it's the DNA's baby. But by process of elimination, because there also is this option that's A, but there's no AB baby available for the DNAs. So I know by process of elimination that it has to be the other couple's baby. So when we were talking about blood type, we talked about the idea of having multiple alleles. So just to reiterate with that, that means that you have more than 2 alleles for one gene. So when we were talking about blood type, we had 3 alleles. We had the A allele, the B allele, and the I allele for blood type O. And another example of that is fur color in rabbits. So let's look at this. I had a hard time with subscripts and superscripts here, so I've just underlined the thing that's supposed to be the superscript in the prompt here. Anyway, what are the possible fur colors of the offspring of a cross between a dark gray rabbit, which has the genotype of an uppercase C and a lowercase c with a CH, and a Himalayan rabbit that has a genotype of lowercase c and a lowercase h superscript and a lowercase c? So this means absolutely nothing to me. I have to come over here and look at the key in order for this to mean anything, which is great. So I'm going to fill this out first, because I can do that without really understanding what I'm writing per se. And it gives me these 4 options. So all I need to do is I just need to hunt for these options on the phenotype key to know what these are. So with this one, I see that this matches this option, which is under full color. For this one, I see that it matches this option for full color. Then for this one, looks like it's this option for chinchilla fur coloration, and then for this one, it looks like this option for chinchilla fur. So that tells me that I have a 50% or 1/2 chance of having a full color rabbit here, or a 50% chance of having the chinchilla fur rabbit. Which these are just fur coloration types in rabbit coats. Fur color meaning that they are a solid color, albino meaning that they completely lack any color along in their eyes. Himalayan rabbits and chinchilla rabbits are like a combination in between where they have some white spots but they're not completely a solid color. Okay, up next is polygenic inheritance. So my prefix poly means many. Genic is referring to genes here. So this is a trait that's produced by 2 or more genes. So it's really important that you understand 2 or more genes. This is different than multiple alleles. You have to know the difference between gene and allele to understand this. So remember, alleles are versions of a gene. So that's saying that it's still fur color, but I had 4 options for fur color. Okay? This is saying that there's more than one gene that's controlling this. So this means that it's going to produce a wide range of phenotypes. So there's going to be like a huge option of different phenotypes. So for example, skin color, eye color, height, personality. If you look at a makeup aisle, you know that there's not just 3 options of skin tone. There's, hundreds of options of skin tone. And that's because it's polygenic. So it's going to create multiple mini options. And because you have 2 genes that are interacting together, something called epistasis can happen. And this is when you have a gene that can overshadow another gene. So for example, different like Labrador Retriever fur colors can be caused by epistasis. That's how you get yellow labs, because yellow labs are just like where they don't have any color in their fur at all. And then also albinism. So it means that something has an albino coloration, which means a lack of color. So there's one gene that codes for yes color or no color, and then the second gene codes for what color. And so if that first gene says no color, then it overshadows what color, right? Because in order to be a color, you would have to say yes to color. All right, moving on to linked genes. Linked genes mean that they are physically located close on the same chromosome, and so they're likely to be inherited together. So for example, blonde hair and blue eyes are typically linked genes. That's why you see a lot of people who have that combination. Red hair and freckles, once again, linked genes, you're likely to see that combination. Large ears and a broad nose, same deal. So linked genes can only be separated or broken apart during crossing over, during meiosis. And since they are so close together, it is less likely to happen. So if you look at the picture here, we have 3 genes on this chromosome that are highlighted. You have the red gene, the blue gene, and the purple gene. The red gene and the blue gene are what would be considered linked because they are closer together on the chromosome than to the purple one. Then you have X-linked traits. So we know that females inherit X-linked genes as normal, and the principle of dominance applies because we know that females have 2 X chromosomes. Females are XX. So they carry 2 copies of this X gene. That means that also you can have females being carriers of genes, which means that it's someone who carries the recessive trait, but they don't show it because they have a dominant trait to mask it. So females can be heterozygous for an X-linked trait because they have two X chromosomes. Males, however, only have one X chromosome because their sex chromosomes are XY. So this means because they only have one X chromosome, they have to express whatever trait it is on their X chromosome, regardless of if it's dominant or if it's recessive. So let's look at this picture really quickly. Remember your sex chromosomes. If you are biologically male, you are XY. If you are biologically female, you are XX. And the superscript is the actual gene that's located on that chromosome, the allele that's on the chromosome here. So in this case, the father is notated XY. Those are my sex chromosomes. And then he has this superscript of a capital R on his X. So this is saying that this is a sex-linked, X-linked trait, and that he has the dominant trait here. The mom, on the other hand, is XX because she's biologically female, and she has a superscript of one capital R and one lowercase r. So this is saying that she's a carrier for this trait because she has one dominant allele and one recessive allele. And this one overshadows this one because we know the law of dominance, that's how that works. So you have different options for these kiddos, which we will show in a Punnett square in a minute. But it is showing the different options that you have. So this would be a normal son because he has the dominant R on his X chromosome. This son is affected by whatever trait this is because he inherited the recessive allele on his X chromosome. This daughter is not a carrier because she got two dominant R's. This daughter is a carrier because she got a recessive R. So let's look at what that looks like as far as doing a word problem and in a Punnett square. So color blindness is a sex-linked recessive trait located on the X chromosome. Cool. Normal vision and color blindness. So this is just showing us color blindness is recessive and it's on the X chromosome. Normal is dominant on the X chromosome. The Y chromosome does not have the gene because it is X-linked. So cross a woman who is a carrier. So woman, she has two X chromosomes. And carrier, that means that she is heterozygous for this. With a man, so his sex chromosomes, XY, who is normal. And so that means his X will be dominant. I don't put anything on the Y because this is an X-linked trait. Alright, so I put my parents on the sides of my Punnett square here, just like I've been doing in all my other Punnett squares. It doesn't matter if you put mom on the top or mom on the side. Same thing. And then I fill out this Punnett square. So whenever I look at this, these two boxes are showing their daughters. These two boxes are showing their sons. So as far as their children goes, only 1 out of 4 children would be colorblind. And that's this one. Okay? But with sex-linked traits, you have to be really careful about what the question's asking you. So if this question is what % of their sons would be colorblind, I'm only looking at these two boxes. So it'd be 1 out of 2 sons are colorblind. If it asked what % of their children are colorblind, then it would be this.